next speaker is Dr. K. Venkat Iyer, Professor of Pathology, AIIMS. Please come, Dr. Venkat.
Thank you. It's a great pleasure to be back at MAMC now. I think this is the first time I'll be presenting a lecture to my own batchmates. I run the cytopathology lab at the All India Institute, and I'm going to be talking about liquid-based cytology and HPV mRNA tests in cervical cancer screening. Now, cervical cancer used to be the commonest cancer worldwide. The surprising thing is that in India, it continues to be the commonest cancer, causing over 65,000 deaths every year. That's the equivalent of having one Hiroshima every year exploded on our country. And so we are all, as cytologists, very passionate about screening and eradicating this disease. We all know that the PAP test was responsible for the great successes which we saw in cervical cancer prevention. And the reason why the PAP test by Papua Nicolau was so successful is because a yearly repeat cytology overcame the problems of poor sensitivity and specificity and of sampling error, which are significant problems of cytology. And the other reason was that cervical cancer takes 10 years to develop. So there was a very long window of time in which this screening could be done. Now, there are lots of problems with conventional cytology, the most important of which, 2/3 of which are because of the sampling errors. So sampling is inadequate, making the screening also inadequate. So in a conventional cytology, what you do is you take the smear, and if this blue is the cancer cell, sometimes it doesn't even get smeared on the slide, and it gets thrown away in the dustbin, and you never screen it properly. So what is liquid-based cytology? Liquid-based cytology is you put the sample which is collected into a fluid and you mix it thoroughly so that the sample which you get has representative cells present within it. And the machine which we are using is called the thin prep. Now the sampling device has also changed. The IO spatula is no longer used. We use the Cervex brush or cytobroom device. And this is far better at collecting the cells from the uterine cervix. And we were the first to start liquid-based cytology at AIIMS. And the technology is very simple, actually. What they do is that they put the liquid containing the cells and spin a cylinder to mix it thoroughly. And in this hollow cylinder, there is a membrane which is fitted on the end. And so you suck the cells and the fluid across this membrane. The cells stick to the membrane. And then when you take the cylinder out, you just press this membrane on the slide, and you get a thin prep smear. And you can, we have done a number of workshops on preparation of this preparation and viewing of these slides. And as you can see, the technique is very simple. You just have to use a sample which is inside this liquid. The slide is kept as a special slide which has a coating on it. And the cylinder spins to mix the cells well. And as you can see, the spinning is very important. It mixes the cells thoroughly so that your sampling is adequate. And once it is inside, the cells are adherent to this surface. The extra fluid is decanted out. And this slide here goes and picks up the cells. And that's all that is to it. So the technology looks very simple, but it's surprising that it took a long while for it to come. And as you can see, once you have liquid-based cytology, you get a far better standardized slide which you can use for screening. And if you look at conventional smear cytology, there's so much of cell overlap, and in liquid-based cytology, you get this beautiful clean smear. So it's much easier to pick up the abnormal cells. And the studies which we conducted showed that the unsatisfactory rate moved from such a high percentage in the conventional smear era to a much more acceptable level after shifting to liquid-based cytology. Now, what is the purpose and aim behind screening, and how do we do it? What are we doing is early diagnosis and treatment of high-grade squamous intraepithelial lesions before it progresses to carcinoma. And if you look at the USA, there are going to be about 300,000 cases of HSIL every year. And that's what we want to screen and prevent. But to be able to detect this, because this is the tip of the pyramid, you also have to pick up the low-grade squamous intraepithelial lesions and the ascus cases. So you have to find 3 million positives to be able to identify the 300,000 who need treatment. So we need something better than just cytology. And as you know, Harald Zurhausen was awarded the Nobel Prize for discovering that it is HPV which causes cervical cancer. And the persistence of an HPV infection results in SYN3, which subsequently becomes an invasive cancer. So the test which came in the last decade for HPV detection was the hybrid capture. This was FDA approved, and this is the technique which we use in our department. So this contains a probe cocktail in which all the high-risk types of HPV are present. And the sampling device is a little different, and it uses a luminometer to run the test. And the technology uses a fairly high-tech device in the form of DNA-RNA hybrids. And that's why it's called hybrid capture. So these are captured on the surface of a well and then tested for its presence using chemiluminescence. So we did a number of studies at AIIMS where we found that although we have so much of cervical cancer in our country, we actually have a much lower rate of HPV infection of around 6 to 8% only. So the reason for the higher presence of cervical cancer in our country is because we have more population. So if you look at the types of HPV infection, there are 3 kinds. One is the latent infection, one is the lytic, and one is the integrative. Now, this is the integrative infection which causes HSIL. It's just 0.5% of all the cases. When it is a lytic or productive infection, it causes LSIL, which gets cured on its own. The problem is that you also have latent infections as high as 8 to 16%, where the virus is there, but there is absolutely no effect of the virus. So currently, the role of HPV testing is changing. In the last decade, we said, is there presence of high-risk HPV infection present in the patient? Currently, our question has changed. We are asking, is there an integrative transforming infection by HPV present in the patient? So what does it mean? It means that we are no longer trying to find out if the viral DNA is present, because even a latent virus will have this. We are trying to find out if the E6, E7 mRNA is being expressed by the virus. And so the type of test has changed. The E6, E7 mRNA, which is required for cervical carcinogenesis, keeps on increasing in the precancerous lesions. And if you look at the tests which test for E6, E7, the sensitivity is much higher than hybrid capture, almost 99.5, virtually 100%. The test which looks for HPV E6, E7 mRNA is also type-specific. So it looks at the type-specific E6 and E7. It has very recently been approved in 2011, and all 14 high-risk types are evaluated. And it uses a fairly complex molecular biology, which I won't go into. And it reduces the screen-positive population to around 4%. So from a situation where 8 to 16 was positive, we now reached just 4%. In the future, we are also going to be seeing a drastic change in techniques of screening. From a situation where we have been looking for the virus, we are probably also going to look at cellular markers. So we are combining the viral and cellular markers so that we are able to screen for this disease, HSIL, rather than just the virus. And of these, the most promising is the methylation-specific PCRs which are coming up. And so the future is expected to move towards methylation-specific PCR. So to conclude, liquid-based cytology is the current standard of care for cervical screening. It improves both sampling and screening efficacy. Hybrid capture for reflex HPV testing is probably going to be replaced by Aptima HPV mRNA testing in the near future. But future molecular biological tests are currently under development, and these are likely to completely replace cytology. Before I conclude, I would also like to give my acknowledgements to my teachers, Professor Kusum Verma and Kusum Kapila. The people who sponsored this talk, that's Hologic, Trivitron, and Vision Diagnostics. And my laboratory, I have two faculty members also working with me. That's Dr. Sandeep here and Dr. Deepali has just joined. And this is my research staff. Mostly they work on Wilms tumor, but they also do the HPV-related work, which we do. But most of all, I would like to acknowledge the importance of Department of Pathology, MAMC, because they are the ones who inspired me to take up pathology, especially Dr. Basu, who I'm sure all of you remember, who is now in JIPMA, last I heard. Dr. Shom, who has now left the department, but I think he's in Gulf now. Dr. Tejinder Singh is still very much there. I keep meeting him in MAMC. He has, I think, recently stopped being head because of some kind of rotating headship. And I also have two of my students currently working in MAMC as assistant professors. So I have a situation where my teacher and my student are both in Department of Pathology, MAMC. Thank you very much.
Thank you, sir. Please wait a moment. May I call Dr. Navratan Surana, please, to come on the stage?
शेयर कर लूँगा वापस दे दूँगा।
Thank you, sir.